Hi everyone. Today we're going to be going more in depth with the DNA by looking at gene regulation. So gene regulation is just the turning on and off of genes in response to the environment. And note that this is very important because you have to be able to adapt to changes in your environment. And so the way that your cells do that is by turning on certain proteins and turning off certain proteins. And just so you know the definition, gene expression is just the process where genes become proteins. And so we're trying to regulate the gene expression. So just to go into examples, the first one we'll look at is in bacteria cells, the lac operon, which is the segment of DNA that codes for lactase, which is an enzyme that breaks down lactose. So as we've discussed, for DNA to be transcribed into RNA, you need the promoter. And the promoter is just the segment of DNA where the RNA polymerase attaches to initiate transcription. There's also something called the operator. And the operator is a segment of DNA that is in between the promoter and the segment, like the nucleotide sequence that codes for the actual enzyme. So it sits right in between. And it's kind of like the control switch that determines whether or not RNA polymerase can actually attach to the promoter. The whole segment of DNA from promoter, operator, and the genes that code for that protein, all of that together is called the operon. And note that this is all for just prokaryotic cells. So here's a picture that shows all of this. You have the promoter and the operator, and then this is the regular lactose gene. And this operator is what determines whether or not RNA polymerase can attach here. So how do you turn off the lac operon? Remember, the reason why you want to regulate is because you don't want to waste energy for the cell to produce these proteins if there's no need for it. So if there's no lactose that needs to be broken down by the cell, then they don't want to create this lactase enzyme, which breaks it down, right? So to turn it off, there's actually something called a repressor. And this is a protein that binds to the operator. And when that protein binds to the operator, it blocks the RNA polymerase from attaching to the promoter, which means that there can be no transcription. And so this repressor, this protein that binds to the operator, it is created by the regulatory gene. And that's just a gene outside of this operon somewhere in the DNA that codes for the repressor. So in terms of turning on the lac operon, clearly you want this enzyme to be produced when there is lactose available. And so when there's the presence of lactose, which is what you're trying to break down, it actually binds to the repressor. And then when it binds to the repressor, it inactivates the repressor, and the repressor will fall off of the operon, which allows RNA polymerase to attach to the promoter and then continue with transcription. And so this is a form of something called an inducible operon, which just means that this operon is normally turned off. In the normal state, the repressor will be attached to the operator, and it's only when there's a presence of lactose available that the repressor will be taken off, and then the gene can turn on. There's also something that kind of like the opposite example, where the gene is normally turned on and you must inactivate it. And so this example is the tryptophan operon, TRP operon, which is called a repressor operon. And so this is an example inside of bacteria cells where tryptophan is an amino acid. And so normally bacteria cells will produce their own tryptophan. So they'll produce it normally. That's what happens. But if there is the presence of tryptophan in their environment, then there's no need for them to produce their own. So then they'll want to inactivate the gene. So in this case, when there's a presence of tryptophan, it actually attaches to the repressor and activates the repressor and allows and prevents transcription from happening. So this is the opposite of lactose, where in the lac operon, the presence of lactose actually inactivates the repressor and makes the repressor fall off. In this case with tryptophan, normally the gene doesn't have the repressor on it, but when you have tryptophan, that presence of that in the environment will activate the repressor and then turn off transcription. Just to note, like we've said, repressors, there's two different types, the kind that is inactivated by the environment or the kind that is activated by the environment. But you can also have something called activators. And activators are just what they sound like. They're proteins that turn on operons by binding to DNA and stimulating gene expression. Okay, so this is now we're taking a step back into a different form of regulation. And just note that cells, they need to differentiate. So your heart cells do a very specific function versus your muscle cells in your legs versus your stomach cells. And the way that your cells can differentiate in this very broad way is by turning on a massive set of genes versus turning off a massive set of genes. And the easiest way to do that is through chromosome regulation. We've talked a little bit about this, but DNA can be packed. And the way it does that is there are these positive charged proteins called histones, and your DNA will wrap around these histones inside of your nucleus. And this complex of the histone plus the DNA wrapped around it, this little thing here, is called the nucleosome. And so, in general, when DNA is wrapped around these histones to form nucleosomes, this blocks RNA transcription because the RNA polymerase can't really read it. And so, when you're packing the DNA, that DNA is not being read. So you can actually do chemical modifications that will either increase packing or decrease packing. Methylation is the process where you add a methyl group to the histone protein, and when you add that, it actually compacts the DNA and makes it much more condensed and much harder to transcribe. And so this is used for long-term inactivation of genes. And so if you don't want any of these genes to be expressed pretty much for the lifetime of that cell, you can methylate it and it will prevent it from being read. It's kind of the same as like putting a lock on a suitcase, and you don't really open up that suitcase unless you open up the lock. And so what's interesting is that these methylated modifications can be inherited. So if your mother has a certain set of genes that are a better example would be like if one of your cells has genes that are methylated, then every time that cell divides and has further daughter cells, all of those daughter cells will have the same methylations, and so the same set of genes will not be expressed. And this is called epigenetic inheritance. And so this is when the traits are inherited, but they're traits that are affected by these modifications and not by changes in the direct DNA sequence. You can also have acetylation, and that's just when you add a different chemical, which is called an acetyl group, and that does the opposite of methylation, and it just opens up the DNA and allows transcription to happen. The last kind of chromosome modification you can have is X chromosome inactivation. And so clearly with females who have two X chromosomes, only one of those X chromosomes is really expressed in each particular cell. And so when that cell, during embryonic development, one of those X chromosomes will be inactivated, and that inactivated X chromosome is called a Barr body. And so whenever that daughter cell divides, the subsequent daughter cells will have the same X chromosome that's inactivated. And there's a picture here of a tortoiseshell cat because here some of the X cells are inactivated, which leads to expression of orange coat, but in the other cells, the other X chromosome is inactivated, leading to black color. Okay, so eukaryotic transcription regulation is a little bit more complex than prokaryotic. And in prokaryotic, we talked about how transcription is really related to the operon, and you pretty much have just a promoter and the operator. For eukaryotic, you have a lot of other sequences of DNA that can control the expression of genes, and these different sequences may not be near the actual gene that codes for a protein. So, for example, you can have something called enhancers, which are just segments of DNA that are located far away from the actual gene itself that codes for the protein. And activators bind to these enhancers, and that's what helps transcription to occur. And so you'll have enhancers over here, which are far away, and they get attached, activators get attached to them. And then there's like a DNA bending protein, and that like helps these genes, these enhancers that are so far away, to bend forward and get kind of close to where it actually needs to be. And when all these transcription factors, which are proteins that help RNA polymerase, when all of these transcription factors come together, the activators, certain repressors, all these things come together, then RNA polymerase can actually help transcribe. So for eukaryotic cells, as you can imagine, it's just much more complex. Here's an in-depth look. And you can imagine that if this was not wrapped around and you laid it flat, the DNA, then the enhancer segment of DNA would be very far away from the actual gene A that you want to express. Okay, so after transcription initiation, which is probably the most important part of regulation of genes, you can also have different alternative RNA splicing. This shouldn't be completely new. This just means that when you take your pre-mRNA, you have a mix of exons and introns. And just like that extra credit question on your test, not all of these exons have to be used. So some of the exons can be used to produce a certain kind of protein, and other exons can be used to produce different kinds of proteins. And this is another way that you can regulate gene expression and just diversify the amount of proteins you can make from one segment of DNA. There's also something called small RNAs. These are actually non-protein coding RNAs. So what that means is for a long time, there were these large stretches of DNA that people didn't think was actually useful because it didn't produce proteins. Now they are finding that you can transcribe that DNA to produce RNA, and even though that RNA doesn't produce a protein, they could be something called microRNAs. miRNAs or microRNAs are just small segments of these RNAs, and they're complementary to other mRNA molecules, and so they can complementary base pair with the mRNA and either have that mRNA be degraded or block that mRNA from being translated. And so these small RNAs are a really important way to kind of regulate what you want to actually be expressed. And if you're producing too much mRNA and your body realizes it wants to degrade them, it can actually use these RNA sequences to bind to the mRNAs and kind of inactivate them. And so this process of purposely injecting miRNAs or microRNAs into the cell to turn off the gene, that's called RNA interference. And the person who discovered this actually won the Nobel Prize in 2006. This is just a picture. You can see that this is the micro RNA that gets coded. And then you can have this microRNA, which is very short sequences, kind of base pair with a regular big mRNA. And then the cell will know to degrade it. Or it can just bind somewhere else and then block the translation of that mRNA. Okay. After all the regulation with in transcription, you can also have regulation in terms of translation. One of them is just the breakdown of mRNA. So after mRNA leaves the nucleus and goes into the cytosol, that can be degraded by enzymes. You can also block or you can regulate the initiation of translation. There are certain proteins that are needed for translation to occur, and so they have to be present for the start of polypeptide synthesis. You can also, after the protein is created, you need to activate some of those proteins. And so sometimes after translation, proteins will need alterations to them, like they need to be cut or they need to have things added to them for them to be fully functional. And that's another way your body can regulate whether or not they want those proteins to be activated. And the final thing is protein breakdown. And so even after you create a fully functional protein, your body can choose to break down those proteins in case you've created too much. So just as a final review, I know that this is a lot of information, which we'll go over again in class, but you can have on the largest scale gene regulation is chromosome modification, and this is when you're inactivating large segments of DNA through methylation. You can activate transcription through acetylation, and you can completely shut down a whole entire chromosome through X chromosome inactivation. Then during transcription, you have a regulation of the initiation process, of whether or not you actually want to start transcribing. In prokaryotic cells, you have these operons, and you can use repressors or activators to turn the operons on and off. In eukaryotic cells, you have just a very complex system which really relies on transcription factors. Those are those proteins that will eventually allow RNA polymerase to transcribe or to not transcribe. After transcription, but while you're still in the nucleus, you can modify the mRNA you've created by RNA splicing, so just taking different segments of exons, or you can use RNAs, which are microRNAs, and that targets certain mRNAs to be destroyed or not translated. After that, just as we've discussed, you can have a lot of modifications during translation. You can either break down the mRNAs so that you no longer use them to produce proteins. You can regulate the initiation of translation. So even though you have the mRNA, you may not create the protein from it. You can have protein inactivation. So that's protein activation. So you need to activate the proteins for it to fully work. And so that's another level of modifications. And then you can also break down the proteins. So these are all different ways that your body regulates what kind of proteins you create. Thanks so much for watching, and I'll see you guys in class. Bye.